I first met Kent many, many years ago when I was a nervous master's student at what was then the Society for Conservation Biology conference in Brasilia. And so it's, and I remember just being, oh, Kent, I'm doing, hello. I just wanted to say hello to this very well published author. And so it's gives me great pleasure to introduce Kent today as our Allegis Goslow speaker for the semester. So after I met Kent in Brasilia, I then worked with him through something that we called the Silwood Group. And we were specifically focused on bringing a more systemic and holistic lens and view to evaluation in conservation. And so it's following the work of Donna Meadows, et cetera. Andrew Knight set that up when he was at Peril College. And through that, then I got to know Kent better, and I'm been very privileged to have him as a mentor and someone I can touch base with. And he certainly helped me navigate the complexities and challenges of developing my career as a conservation scientist. So as I said, when I first met Kent, he was working for the Wildlife Conservation Society, where he was the vice president of conservation science and strategy. Before that, he was at the Nature Conservancy, and before that, he was faculty at the University of Florida. And over the past decade or so, he's been working with his own company, Archipelago Consulting, and has been doing some amazing work at the cutting edge of the challenges we face in conservation today. And so he spent 9 years working at the intersection of conservation and synthetic biology, and for 6 years was chair of the IUCN's task force on synthetic biology and conservation. And that complex issue is what he'll be talking about today. Also, in the pandemic, Kent led the GEF, the Global Environmental Facilities, which is the world's biggest chunk of money that goes to environmental conservation. He led the task force on a post-COVID strategy, and there's a paper on it published in Conservation Letters. And also, as some of you may have seen, Kent recently published from his work in synthetic biology and conservation, a book with Bill Adams from Cambridge, Strange Natures, Conservation in the Area of Synthetic Biology. So it gives me great pleasure to hand over to Kent, and welcome.
Thank you. Thank you, Dawn. It's a pleasure and a privilege to be here, and I appreciate the day that you put on for me today outside. So I haven't had a chance to see your fine city yet, but look forward to the weather improving tomorrow. So what I have for you today is a really rapid tour that starts with the way I was raised when thinking about nature and takes us through this era of synthetic biology. And we'll be stopping along the way, and you'll be following my own personal journey as I learned through all of this. So, nature, the Bob Marshall Wilderness in Western Montana. Nature, the Peruvian Amazon. Nature, gorillas. Nature, this is what I worked on for my PhD, giant anteaters. I was raised many years ago to think about nature as something that was entrancing and something that was other than human. And at that stage of belief, nature needed conserving from destruction, and that was destruction by people. Because we all knew at that time that humans were bad for nature, and that saving nature meant removing humans, and then it would thrive without any human interference. At about the stage that I was in graduate school, I began to really have a transformation and to understand that, in fact, this dichotomy was not something that could be sustained. It was not typical of the way the world was, in fact, constructed. A few examples here. Some of you may know lyrebirds are a Southeast Asian bird, which is a superb mimic. And these birds have incorporated into their own vocal repertoire the sound of chainsaws. So, or we see here in the Chinese Panda Raising Center where pandas have been taken from their natural parents and being raised by people dressed up as pandas. Or this particularly evocative and sad photograph, the lack of adequate shells is meant that hermit crabs in some beach settings are now using bottle caps as a way of protecting themselves. So in this intertwined world, these three of many examples, humans have been practicing numerous interventions to maintain naturalness, creating this sort of strange nature. And it's this tension between the natural and human interventions to maintain the natural that I want to illustrate with a set of things that you will regard as really quite normal. So removing invasive species or restoring species. I particularly love this picture because these are parachuting beavers. But you didn't know they knew how to do that, but there they are. Cutting, of course, replanting, of course, predator control, of course, supplemental feeding, this particular amazing photograph from Spain of places for feeding of vultures with the carcasses of domestic livestock. And of course, river restoration here from the state of Maine, where I live, the removal of this dam to create a free flowing river. And of course, burning, which is very much something you in this part of the world are familiar with. So we are increasing using these things which might be called artificial as ways of trying to preserve what we would be thinking about as the natural. But this notion of unnatural nature, which I've illustrated and which has this history within the conservation practice, in fact, is something that does not exist in other parts of the world where such interventions have been baked into long cultural practices. So this is a flooded savanna in Amazonian Bolivia, and you can see the forest island in the background. This is only raised a couple of meters above the flooded area. But in fact, when the archaeology is done, you find that these trees are growing on soil which is almost entirely composed of the remnants of previous human civilizations. Or more recently, some of you may have read these examples from the Northwestern North America, where clam gardens, turns out that people before the Europeans had arrived were actually building structures in order to increase the productivity of these coastal areas for clam production. And of course, the production of forest, the enrichment and the modification of, quote, natural forests in order to increase the percentage of trees which produce fruit that humans desire, this is a Southeast Asian, this is a Bornean photograph in the back, or the actual creation of forests, very rich in medicinal plants, as here in the far eastern portion of the Brazilian Amazon. So all of these examples, and in fact, most of the things that you guys think about, most of us think about in these examples about nature are nature as either ecosystems or species. This is the way we tend to think about this. These are two of the three components of biodiversity. In fact, there is a third component. You all will know that. The sort of classic definition of biodiversity being ecosystem, species, and genes. But this third portion of biodiversity is what I want to talk about for most of the rest of the talk. And it has not been addressed by my colleagues and me within the conservation field for very interesting historical reasons that we can get into if that interests you. So the point I want to be making to you here is that the tools that humans have been using in these strange ways of preserving nature for ecosystems and species are these burning and predator control and replanting, all those things that you think of as kind of natural. They've been normalized. But there is a set of emerging tools which are directed at the genetic component, which are being proposed to be used to conserve this genetic component of biodiversity, which are far from normalized. And there I will be talking about this argument that's going on about this. And the potential use of these tools is forcing us to ask the same questions that we were asking about species and ecosystems, only this time we're asking them about the use at the genetic level. What kind of nature do we want to conserve, and what are we willing to do to conserve it? So these tools are brought to us collectively by this term synthetic biology. This is a book that was published a number of years ago by a colleague. It really sums it up. Biology is technology. And this quote from one of the developers of the field, the genetic code is 3.6 billion years and it's time for a rewrite. What has happened is that the engineers have discovered biology and they have found a system which is in need of engineering help. It's inefficient, it's unproductive, it has a lot of waste, and they know that they can do things better. They can change biology to more directly and efficiently produce things that humans want. So a brief introduction for those of you for whom this might be a new field. So let's think about cows, particularly here in Arizona. You think a lot about cows. So when you think about cows, and you're not talking about the destructive grazing practices, you're thinking about leather and meat and milk, which come from cows, unless they used to come from cows. These are now 3 companies, New Harvest, Perfect Day, and Modern Meadow, that are bringing you meat and milk and leather that never saw a cow. This is being done by taking the genetic component from cows that produce the meat, the milk, and the leather, introducing them into algae, and in large industrial vats, producing these products and marketing them without ever having seen a cow. So what is this field? Now, on your left is the Convention on Biological Diversity's definition. I'm not going to use that, but it's a key thing that we'll return to if you care about the policy portion of this. I find much more useful this definition in the box. Synthetic biology is a scientific field that incorporates biology, chemistry, computer science, and engineering to build biological systems with abilities that they inherently lack in nature. And it can be viewed as redesigning biological systems to perform a specific task with a user-defined purpose, and those users are all humans. So there are a number of ways in which these genetic changes are being made. These are just a few of them: altering DNA outside of the cell and reinserting it into another cell, altering the DNA in the cell itself, or rewriting the DNA and adding nucleotides. And we'll return to this later. The technology which has been most commonly used in the press has been CRISPR, which is an evolving technology. There are lots of different versions of it now, which is a way of making very precise alterations within the genetic code of a living organism. I want to impress upon you the range of this work that's going on. So this is a global map, and it's showing publications. So, and the darker the color, the more publications. The point is to see that it's distributed across the entire world. So the US and Europe used to be the leaders of this. China and other countries like Singapore and Southeast Asia have rapidly taken this up, as have Russia, Brazil, Chile, Argentina, Australia. So it's found across the world. And it's a very rapidly setting change. And I want to take a moment to try to explain these things that are called Carlson curves. Some of you may know about these. So the blue one is what is called Moore's law. And some of you may know about this. This has to do with the decreasing size and the increasing effectiveness of chips, microchips. And that's the technology that's brought you your cell phone. And you're familiar with this often used phrase that there's more computer power in your cell phone than there was on the first lunar lander that went to the moon. And that's the speed up until 2014 or so, at which change happened in that technology. So we're asking the question here, what does that tell us about the kind of change we might see in the genetic technologies? And so basically those two technologies are reading DNA, that is decoding it, and writing it, that is changing the DNA in ways that humans want. You see the red and the yellow curve, the red being reading and the yellow being writing. You see that the rate of change of these technologies is more rapid than it has been for Moore's law, suggesting, if it's an appropriate comparison, and there may be people here who will know better than I, that we can anticipate change that is even more rapid in this application of synthetic biology than we have seen with computing. And it's also very important in national industries. This is old data, 2012, but published in 2016. They're very hard to find, but even in 2012, in the United States, $324 billion worth of gross domestic product was in this biotech. You can see here GM crops, industrial applications, and biologics, that is particularly medicine-related things. And it's not just universities and companies that are doing this. And this is a particularly important thing. I don't know if you're all familiar with the DIY movement. It's the do-it-yourself movement. So this is a conviction that that the ability to manipulate technologies should be community and individually accessible. And so these blue dots are labs where for a small fee, anybody who wants to can walk in off the street and have access to the tools to make the kinds of genetic changes that we have been talking about and will be talking about. Anybody who wants to go in can do that. They're not the most recent iterations of the machines, but they're very contemporary. And so it's not just confined. And it's also not confined. I need this slide because I think of you as young, but in fact, there are people who are younger than you are. So there is a competition called iGEM, which stands for the International Genetically Engineered Machine, which is about getting students to develop organisms, forms of life that have genetically altered. And then they have these jamborees and they bring together thousands of students from across the world. So the yellow curve on the bottom is the number of countries. So it's up to 50 countries that are involved. And the number of teams is the blue. So very broadly, and this started out as a graduate student exercise. It then went to undergraduates. It's now extended to high school students who are engaged in genetically altering forms of life. And because there are younger people and high school students, there are now elementary student kits and books so that this kind of alteration is being done at the elementary school level as well. And in the New Scientist, which is one of my favorite journals, on the flight out here, there was an article about a young man, a high school student in California named Madhuredi, and his angelfish died. And he froze the angelfish from his aquarium. And he went to a community lab and he produced the first genome of the angelfish, which he did on his own money with his own technologies, and then submitted it. And it's now joined the large field of genomes of wild species. The point being, it's very broadly dispersed already. Oops, excuse me. So there are two areas in which this technology is most advanced, where most of the investment is. The second of these two is agriculture. So there are a set of amazingly ambitious promises that are being made and projects that are being developed. The first one is the one that just kind of really spins my head around. So rice is the most important subsistence grain in the world. And it is an inefficient form of photosynthesis, the C3 photosynthesis. And plants that have a more efficient photosynthesis are C4. So there is a big effort now to actually change the rice plant so that it's no longer a C3 and it becomes a C4 plant. And the expectation is that will greatly increase the productivity of rice. But it is changing one of the most important food grains in the world at arguably its most basic level, which is its photosynthesis. So also increasing micronutrients and creating new domestic crops. So there was a new tomato that went from wild relative to domesticated plant in 5 years, unlike the thousands of years that have taken to domesticate plants through directed evolution and synthetic biology. So increasing drought tolerance, increasing disease resistance, increasing nitrogen fixing, and then a whole set of applications with domestic animals that we can talk about if you want to. So just a few of the most recent things in agriculture. So this is came out in The Economist this year. This is a proof of concept to increase soybean, which is extremely important both for its oil, but also as a grain to feed livestock, increasing its productivity 20% through alterations made with these synthetic biology tools. This one is a particularly interesting one. So, some of you know, or yourselves take omega-3 fatty acids, which are called fish oils. In fact, they don't come from fish, they come from algae that the fish consume, but they bioconcentrate in the fish, they're harvested, they're sold as a nutrient supplement. It's a very important part of health. So the the genetic sequence that makes omega-3 fatty acids was taken out of the algae and inserted into canola or rapeseed plants. And so these plants are now being produced, and the oil that is generated from them has this enriched omega-3 fatty acid, thanks to the genetic sequence that came from the algae, which we think about coming from the fish. And then this is a kind of another one of these amazing things. These are poplar plants, poplar trees you're familiar with, that have been genetically altered to much more rapidly sequester carbon. And the idea is to be growing these genetically altered poplars in order to harvest carbon. So carbon becomes the crop, or sequestered, with then buried biochar or other sort of ways. All right, then human health. So this is where the big money is, not surprisingly. And you will probably have read some of these applications. I'll just jump ahead and then return to this. This, of course, is the big one that you'll know about. Any of you who've had 2 of the most common COVID vaccines, those vaccines came to you courtesy of the tools of synthetic biology through mRNA. So they're also being applied to reduce disease burden through malaria, develop new drugs, drug effectiveness, a whole set of amazing proposals for monitoring disease within the human body by modifying bacteria that then serve as monitors, release a chemical into the bloodstream, which can be detected as an early warning sign of disease. And then, of course, improved nutrition. We did the vaccine. Here's another one, you can sort of, these are, if you have roommates and you eat dinner with them, you could talk to them about this. So there's a very interesting fact, which is that pigs have an anatomy. The pig anatomy is the closest of any other species in the world to the human anatomy. And this was known starting in medieval Europe, when it was illegal to operate or to teach on human cadavers. Pigs were used. And so people have known that pigs had very similar systems to humans. The problem is that there are a set of retroviruses in pigs which are embedded into the blood system in the organs. So when those organs were transferred into humans, the humans then got this retrovirus, and it caused rejection of the organs. So George Church in his lab at Harvard modified pigs to eliminate these retroviruses, making pig organs now much more compatible as what are called xenotransplants for humans. And it is anticipated that there are now going to be pig farms being raised specifically to provide organs for transplants into humans. Then this is happening across an incredible range of things. So the anti-cancer drug vinblastine is produced from a plant, and the efficiency of the extraction of the 2 precursors of this drug is very low. And so the medical practice has not been able to get its hands on enough vinblastine to be able to use to prescribe. So, of course, this is the thing the engineers love. I know how to fix that. And they remove the genetic sequence that make these 2 precursors, put them into algae. Now this vinblastine can be produced in industrial vats and is much, or will be, we hope, much more accessible to be used in cancer treatments. So agriculture, wow, human medicine, OMG, you got to ask, if you're me, when you read all this stuff, well, what about us? What about those of us who think about and worry about the natural world? That is not a topic that is on the minds of synthetic biologists at all. They're busy founding companies, raising money, startups, publishing Nature papers, doing all of this great stuff we talk about. So over the last number of years, really starting in 2013, I've been involved in helping set up and run a set of meetings to try and get the synthetic biology community to understand the potential, some of the potential applications that those technologies have in the conservation world, but also some of the deep concerns that those of us in the conservation world have with them screwing around with this 3rd component of biodiversity. This last one, the Marseille meeting there in November 2021, that'll be featuring later in this talk. So I have a lot to say about that. So I was asked to put together a group of people by IUCN, which is the International Union for the Conservation of Nature and is the world's largest environmental organization. So we did this assessment of the potential applications of synthetic biology to conservation. And I stress potential, because as far as I know, none of these things that I'm going to be talking to you about have actually been done in the wild. They have been proposed, they have been tested in lab settings, and in some cases in kind of large cages. So this is available. We looked at a set of these proposed applications, and I'm going to walk you through these. So one of the things synthetic biology has enabled is the creation of a technology called engineered gene drive. Gene drives are naturally occurring ways of overcoming sexual reassortment. So it allows you to hook a gene that you are desiring to force into the whole population to a genetic sequence that causes it to be fixed in a population at much higher than Mendelian rates. So there is a project, a big project that it's called GBIRD, that is a consortium of US and Australian and New Zealand institutions to use this gene drive mechanism in invasive house mice on islands where there are predators on seabirds, and to create a mouse that will only have male offspring. So there will be no killing of mice, but there will be a gradual extinction of the population, and therefore the loss of that stress, their threat to these seabird islands, to the seabirds. Then this is a particularly interesting one. You may know that there is a mass extinction of native birds on Hawaii, and one of the major threats and stresses on them has been avian malaria, which is an invasive disease carried by an invasive mosquito. And so there is a real interest in trying to create a way to prevent this avian malaria from being transmitted by these mosquitoes. This is a really interesting one, but it's actually moved quite far. You may know black-footed ferrets are one of the most endangered mammals in the US. And so it's been reduced to a very small number of individuals in the wild. There's a huge effort to try to build up the population and release them. The problem is that there were only 16 individuals that were used as the initial breeding population. And there were a set of frozen cells that were not represented in these founding populations. So they have now succeeded in being able to take the novel genetic variation out of these frozen cells and putting them into a live black-footed ferret to allow it to be reintroduced into the population to increase the genetic variability within the wild population. This is one particularly fun. So that's a photograph of American chestnuts, the way they used to be. And you'll notice the two people standing there. So this was what some people have claimed was the most common tree in Eastern North American forests, particularly in the central and southern portion of the East Coast. And they were huge, and they produced these nuts. So chestnuts roasting on an open fire, we're coming up to that season. Those were American chestnuts, and the nuts that were produced were absolutely prolific. Some argue that they were the major food for passenger pigeons. At any rate, they were a major food for humans and many other animals. In 1904, in the Bronx Zoo in New York, the first detection was made of Chinese chestnut blight. So the American chestnut is a species within the genus Castanea, which is found around the world. There is a fungal blight which is found in the Chinese version. These trees were brought in because they have a shorter generation time. People didn't want to wait that long to get mature trees. So they imported the Chinese in order to produce a faster nut. The fungus hopped on to the American chestnut. And within a matter of decades, the estimate is that between 3 and 4 billion trees died as a result of this fungal blight. This fungal blight causes a change in the pH within the tree which enables the fungus to expand and either kills the tree, but it kills the tree, but it'll leave a root growth so that they can resprout, but they can't mature. The people at SUNY Stony Brook, ESF in New York, figured out that you could take a gene from wheat. It's also found in moss and sugarcane and a whole set of other things. You could move that gene into the genome of American chestnut, and it was largely immune to the blight. So this tree, there's a petition in front of the US Department of Agriculture now to be able to allow outplanting of these genetically altered chestnut trees with the potential to put them back into a system where they were missing. This is at a much more speculative phase, but in my opinion, it's by far the most important potential application. You're all familiar with the horrible plight of reef-building corals, particularly tropical ones, both because of warming oceans and also because of increasing acidification. And so you're familiar with the zooxanthellae, the algae that live inside the coral. They are expelled, and that's what causes coral bleaching. So there's a set of really interesting work going on now to try to alter the genome of the reef-building corals themselves, or of the algae that live within them and that produce the nutrients that help support them, in order to allow them to survive at a greater level within warming and more acidifying oceans, with a potential enormous benefit for humans and, of course, for biodiversity. And this is one of these, oh my God, yuck, but if you don't know this story, you need to know this story. So these are horseshoe crabs, which have been caught, they're alive, and they're being bled. There is a chemical within the blood, the blue blood of horseshoe crabs, which is an absolutely essential component to test for contamination within all the vaccines that humans use. That is where that chemical comes from, the blood of horseshoe crabs. There is now a chemical that has been developed on the market through synthetic biology, which does not use horseshoe crabs, but could replace this if market forces and society allowed it. And some of you may know this story. Horseshoe crabs are not only important because they're horseshoe crabs, but because they do these mass spawning events, particularly in the Delaware Bay and the eastern US. And their eggs are an absolutely critical nutrition source for migrating shorebirds, particularly red nuts, who feast on these eggs, fatten up, and fly all the way to Argentina. This population of horseshoe crabs has diminished, I don't know, 90% or something, because of the harvest for their blood and also for crab bait. This is a complicated one, and I'm only going to touch on it very briefly. This is the same gene drive technology. This work is progressed really quite far to develop a mosquito which will produce, when it breeds, the genetically altered mosquito is a male, and when it breeds with wild females, the female will produce only male offspring. So that's the theory, and the idea is this is the major source of child mortality in all of sub-Saharan Africa and is a problem everywhere. And this is an approach that these folks have been working on to try to deal with that. You say, well, that's not a conservation purpose. It's a very interesting causal chain that you can think about what might happen if you eliminated or substantially reduced the major source of child mortality in the continent with the highest rate of population growth. Speculation, but I think it has conservation benefits. Assessing honeybee collapse. So you're all familiar, some of us are bee experts here. So you're all familiar with the colony collapse disorder and all of these issues that have been confronting honeybees, particularly commercial honeybees. And there is work that is going on now. And in fact, I included this box here because just this month, this company, Dellen Animal Health, announced that it has developed through synthetic biology the very first vaccine for honeybees against this particular fungal disease, foul brood. And because I know you guys have a microbiome center, and because I am the world's biggest fan of microbiomes who actually doesn't know a lot about it. Why are you laughing? There's a lot I don't know a lot about. So everybody knows about microbiomes, I assume. You know, there are, if you look, when you woke up this morning, if you looked in the mirror, looking back at you were more non-human cells than human cells. There is more DNA in your microbiome than in you. So you are not an individual. You actually are an ecosystem, you and your microbiome. So all animals and all plants, and in fact, many microbes have their own microbiome. And so it may very well be that some of the important conservation advances that might happen through synthetic biology might not involve modification of the host genome, but of the genomes of elements of the microbiome. This work is very aggressively going on in humans, and there's no reason that it couldn't be considered, particularly in folivorous leaf-eating animals like this colobus. So, okay, so we did this assessment. We assembled this group of people from around the world. We spent 2.5 years evaluating all this, looking at the evidence, weighing it, writing it up, it's showing what the potential benefits, what the potential downfalls are, and then presented it, the first draft of it, at this Convention on Biological Diversity Conference of the Parties, which is when all the countries that sign this convention meet. It was in Egypt, in Sharm el-Sheikh, the same place that the global climate COP is going on right now. And we just got slammed by people who hated the exercise and hated me and the conclusions and the approach that we took. There is a clear understanding that we laid out that is really important for you to remember, and I've said it once, and I'm going to say it more than once, which is that almost all of these applications are theoretical or early in development. It is, nobody has asked for permission, with the exception of these chestnut folks, to actually do this in the wild. It doesn't matter. These questions, these issues are so deeply rooted in the values and worldviews that people hold that they are, the people who don't like it are absolutely just crazy about the potential, as are some of the people who are crazy about the potential, but on the other side. So the idea was that this technical assessment, it was, right, and there is a whole one, and his colleagues are involved in the whole science policy interface. Here is, this was meant to be, that arrow was meant to be science leading to policy. So we did the science, we evaluated it in the most transparent fashion. We published a first draft. We received, it went out to 10,000 email addresses. We invited line by line, word by word comments. We recorded each one of those comments. We recorded our response to each one of those comments when it was republished. And when I went to Marseille last November to present this, man, it was dirty political fighting. It was the most extraordinary event I have ever been a part of. And members who are against this, they're particularly European groups, got themselves appointed because they're not members of IUCN, so they couldn't formally be in the meeting. They got appointed to be members of the delegations of members who were there, and then systematically and very effectively sabotaged that effort. So we ended up, you got to love this. So this was a 5 years worth of work and probably $350,000. And the decision in the end, there were 4 votes that were taken. We lost 3 of them. We only won 1. And the one we won, which is that they wouldn't, the members decided they weren't going to reject the whole thing, but they were going to, wait for it, establish another working group and give them 4 years to do exactly the damn thing, the same charge that we did 4 years ago. So, and I got told that that was a success because it wasn't rejected. So it was at that stage that I resigned and said I wasn't going to have anything to do with it, but my name was mud in this situation anyway. So science to policy. Okay, now there are a set of really interesting ongoing issues that are this science policy interface. And I'm going to go through these really rapidly. They span the front, but it is in these places, it is on these topics that policy was voted down. It's not the science. So let's go through them. An economy of promises. This is a fabulous notion that Quet published. There is a massive production of scientific promises now. In order to raise money to get the research done, scientists are promising things. Those promises then enter the public discourse, and that becomes, so it's about the promises that were made much more than the technology. Here's a good one. Nature services will be synthesized. I don't need a native forest to sequester carbon because I can sell the wood. I can cut the native forest, sell it for wood. Then I can plant these poplars and I got more carbon sequestered than you would out of the native forest. And I made money by selling the wood. Kind of worried to some people. This is a big one. We're going to release these things out and then evolution's going to say, hello, I haven't met you before. I got plans for you. And this is particularly the case with things like this because white nose is a fungal disease of bats you may know about. It started, anyway, it has spread extremely widely through these hibernating bats and it wakes them up in the middle of winter and then they start to fly about, they can't find food and they die of starvation. If you're going to try to address white nose syndrome with synthetic biology, you must have a self-dispersing organism, which only makes worse this concern about meeting evolution. Okay, privatization. So these are two graphs. The red is publications and the blue is patents. So almost all of these technologies are being patented. So what happens with this black-footed ferret, right? If I patent the sequence that I took from that frozen egg and put it in an endangered species and release it into the wild, who owns that? This is the kind of thing that we're going to have to be worrying about in this regard. So land use impacts. So does anybody know, this is from Hawaii. Does anybody know what those things are in the photograph in the middle? Those are algal raceways. So if you are getting blue-green algae put in your smoothie, it is being grown in something like that. In order to grow algae, you need to have shallow water that moves rapidly because they need to be able to photosynthesize. That's on Hawaii. Think about the water use in that. So the background is oil palm plantations. So there is now released a form of palm oil, which is made by algae. So you don't need palm oil plantations anymore. Hallelujah, save the forest, except, wait a minute, where are we going to grow the algae? If we're going to take that, then maybe we would be better off with the palm oil plantations. So we got to worry about that. This is one of the major sources of concern by people who don't like the agricultural applications of synthetic biology is because of the really unknown implications for really dispossessed and poor subsistence farmers. Then here we go, back again. There are some people who just hate this. So extremely effective communicators, this ETC group. Look at that, driving under the influence. This is a critique of gene drives or extreme genetic engineering. So there are NGOs and organizations whose entire raison d'etre is to be against these technologies. We've all voted yesterday, or those of us who can vote. So you will have seen this figure. So this is the vaccination rate versus who you voted for in the last election. So higher Trump votes have 44% and higher Biden votes have 80%. This is the politicization of science and knowledge. So not getting a vaccine became a political act. It wasn't about the science. My own field, those of you who are in conservation have got this bummer problem, which got itself an official name, last chance tourism. You can look that up. You can go on tours to do one of these two things. This is the last chance you'll have to see these species before they go extinct. This is the last chance you get to go see Greenland glaciers before they melt. How screwed up is that? Last chance tourism. So then, as if we ever had anything in terms of financial and social support. So $2.8 billion 2021 National Park Service request, 2.8 billion, 10 billion ice cream, and 100 billion on vet services. So that's what we're dealing with in terms of a larger context. I love this ice cream notion. I mean, and probably much of it's sold in the national parks anyway. So, and then we have this issue, which is kind of for me one of the most interesting and complex. You're all familiar with the argument about wilderness, that it's a social construct. As Gary Nabhan wrote, conservationists had to create the wilderness before they could save it. That in fact, the Yellowstone model was the exclusion of indigenous people in order to save the natural ecosystems. And it's largely discredited, but it's still an important part of the discourse. It has now been transferred, in my opinion, to the genome. So these people who don't like synthetic biology say we have no business playing God, and that's the language that's used, by messing with genomes. As if humans haven't been messing with genomes since we invented agriculture, and probably well ahead of time. So much to talk about there. Then this one, I love this. We think, and we're raised in a university, that there's one door in your brain called fact, and one door called fiction. And I'm going to class, and I'm going to write all these notes. I'm going to have to write my homework and file it in the next day. Then I'm going to go and I'm going to watch some TV show. And that's going to be the TV show. Only what I've learned, what I've learned over this while, there are two doors, one marked fact, one marked fiction. You know what? They open into the same room. So the most common thing over 8 years of giving public talks has been, my God, I can't believe you're actually talking about this. Have you not seen Jurassic Park or read Frankenstein? And I say, I have watched and read, and they're works of fiction, and it makes no difference at all to people. So, because I'm a huge fan of this gentleman, and I really recommend you see this movie introduced CRISPR to the American audience, because there is a rogue scientist who has a space station. There's an accident and a giant rat that was made with CRISPR breaks it out, and these capsules of CRISPR descend and create these giant animals. And of course, who but Dwayne? We have to save us. That is how people are learning about synthetic biology. Is that not a problem for us? I think. Anyway, I think it's a problem. So we're winding up. We learn in this world of strange nature, which is why we called the book that. I mean, look at this guy. He's vacuuming a whale. And how are we supposed to respond to this? So we are continuing to act as if this nature that we now value can survive without us, while we're simultaneously engaged in these types of management that are becoming increasingly strange. Our daughter lives in the Netherlands, and the wolves have come into the Netherlands on their own without being reintroduced. The Dutch are now so worried about the wolves that they have given the army, a special unit of the army, paintball guns. And they are stalking wild wolves and shooting them with paintballs to make them wild. I mean, if that isn't strange, I don't know what is. And of course, you all know the national color. If you don't, I mean, they're going to be winning the World Cup. It's bright orange. So I'm sure all the wolves are going to be bright orange too. So this is a whirlwind. So where does this leave us with the rise of synthetic biology and its potential applications? So this was published by one of these, the German organizations that was a particular antagonist in this IUCN. And I want to read this quote. Along with its diversity, beauty, and utility, nature's uniqueness is a protected good in nature conservation that is firmly embedded in society. Its intrinsic value imposes limits on the extent to which humans can intervene in nature to protect it. If permanent, far-reaching, and inheritable genetic modifications of wild organisms, if they become a reality and are accepted as legitimate instruments, the idea of protecting nature turns into the idea of redesigning nature, which would limit the value. That, in three sentences, is the objection to this entire enterprise. To me, it is a complete misreading of the state of nature and what we are going to need to be needing to do to try and conserve at least some versions of it. So this is a key point here. Until now, or until the last short while, every living thing on Earth descended directly from some other living thing. That's not true anymore. But that's a huge deal. Now, part of that has been brought to us by synthetic biology, and it's not going away. Look at that, $1 trillion is the global market size last year. Now, you know, I get accused of throwing flash grenades, and so I apologize if this is another one, but this is just too good. So you all know, because you're smart folks and have done this, that there are 4 nucleotides that make up the code of life, and DNA underlies all living things, and there are 4 of them, except there are now not 4. In 2014, 2 new nucleotides were added. 2019, there were 4 new ones. And this year, 7 new nucleotides. So DNA, there is DNA now. That is DNA that has 11 nucleotides and not 4. And you know what those 7 are going to be used for? They're going to be programmed by engineers to produce things that nature has never produced. So this is Belphegor, who is one of the 7 princes of hell. And the reason to talk about him as I'm winding up is that this concern about technology and the role of invention is not a new one, but it is so deeply embedded, at least in Western cultures, that it got its own prince of hell. And he's the demon of sloth and the demon of discoveries, who helps people make discoveries and seduces them with these new inventions that are destined to fail. This is the culturally embedded story and concern about new technologies. We must respect it, and we must think about where our position is going to be in regard to Belphegor. And, you know, because we're being recorded, I won't tell you, but it's important that you look at that disc and what he's sitting on. So synthetic biology has this potential to be a disruptive technology for conservation. It already is. You should be asking yourself, oh my God, I never heard about that. And that's exactly why I want to give this talk. Forcing us to face conflicting objectives, values, and the datedness of much of our thinking. So this is 3 slides left. What I know is that conservation is not going to stop the deployment of synthetic biology. There is this hubris we have that society will not do things if we, like a 3-year-old, sit down and have a fit and say, no, you can't make me. It's going to happen. But we can shape it, but only if we're willing to engage in this. And this is one of my heroes, Sheila Jasanoff. She has written about this thing called a technologies of humility, and this is where I stand. This is the last slide. Methods, or better yet, institutionalized habits of thought that try to come to grips with the ragged fringes of human understanding, the unknown, the uncertain, the ambiguous, and the uncontrollable, that acknowledge the limits of prediction and control. These technologies of humility confront head-on the normative implications of our lack of perfect foresight. Thank you. This is a competition that Lego had at the Bronx Zoo when I worked there to create the Lego giraffe.
Well, thank you, Kent. That was very entertaining and informative and raised a lot of questions in my mind. So we have a couple of minutes for questions before we have some little snacks here, which I was suggested not to put outside because someone would steal them. So they're down here. But so questions? Yes.
The reason is that Mus musculus, which is the house mouse, is native to Europe, and the concern is that if this gene drive were to escape, that it might lead to the extinction of native populations of house mice. So if you buy me a drink, we can talk about the word real. I mean, if it's effective, if they are able to develop this, and if it's deployed, and if it works, then, and if such animals were transferred to Europe and were able to establish a breeding population, you could lead to a substantial diminishment in the native populations, if not extinction of those populations in Europe.
Question over there.
Flowing like poison synthetically themselves, and I think Gensel made in the humane way where you don't have to killing the animals. Why are they using pig poison instead of just throwing them themselves? I love people. And that's vegan, by the way, so animal rights is important.
So, and the argument is made for some of the work in domestic animals that in fact there's a substantial animal welfare component to it. I am not an expert in organ growth, but I don't think that any of it has reached the stage where an organ that is actually suitable for transplant has been created in a lab. But I know that people are certainly working on trying to do that sort of thing.
Are there any questions on Zoom, perhaps? None?
There's a gentleman there.
Oh.
Yeah, I guess a couple questions. One, so is the main issue that you kind of face in that there's no nuance of this, and that we can't even discuss these topics in terms of using gene drive technologies to wipe out things like mosquitoes that might be based on islands? Because I mean, you raised some very legitimate concerns in terms of things like biological control. Australia is a really good example of an issue where unintended consequences have caused problems. And so I guess that would be one of my questions. And then the other concern that I think makes sense to me in a way is that when you go, not to say you cannot alter genomes, but when you're starting to change things, species that have taken millions and millions of years to evolve, that's kind of like, that's our natural history there. And in a way, I can see needs, conservation needs for changing genomes to preserve species. But at a certain point, when you continue to alter these things, you're altering our evolutionary history. So I mean, I know those are two very different kind of questions.
Sure, of course, no, good. The problem is always that people who take extreme positions tend to define the nature of the interaction. So, and most people haven't thought about this, but when there are people who hold very strong positions for and against, it's easier to just agree with somebody than it is to have to think about it yourself. And if I ask one thing of you, I ask of you, I've given you this, I ask that you think about this and that you form your own opinion. I'm not telling you, I don't want to tell you what your opinion should be. What I want to tell you is you better have an opinion, because if not, it's going to be the usual conservation thing, which is we get hit. My image is that conservation is sitting in a bus looking at the back window and always surprised when new stuff happens. I don't want you to be surprised. This is going to happen. So form an opinion. So there's a real polarization of the pro and the con. So it's called, this economy of promises and perils. This group, or at least a major portion of you, is the group that should be thinking about it and coming up with your own opinions. And then you're going to be involved in careers or voting or whatever it is you're going to do that are going to influence that. So that's how I deal with the first one. The second one, does everybody know what counterfactuals are? It's the question of what would happen if you did do something compared with what would happen if you didn't do something. And I think humans are very bad at making that comparison. We all want to say, how could you possibly think about doing that? Dating him, drinking that, you know, choosing this, going whenever. But you have to be asking, what would I do if I didn't make that choice? And that's what I think the conservation and larger society needs to say. Biodiversity is going down the tubes really rapidly. And so it's not a question of whether or not synthetic biology is good or bad. It's a question of whether or not we have any other choices, or we're going to be placed in a position that we're either going to say, as these Germans would say, we're not going to touch it, and it may go extinct, but at least we didn't touch it. Or we're going to say, we touched it, we altered it, but in altering it, we have engaged as an evolutionary actor, which we have always been. I mean, that's what the climate change issue, that we have become the world's most important, as a species, the world's most significant evolutionary actor. It's not like it's all fine and then you're going to screw it up with coral genes. It's all screwed up. And then what are we going to do about it? And that's, so that to me, that's the question that has to be asked. And when framed that way, I don't know, you're going to have to decide what you want to do, what you want to support about that.
Thank you, Kent. Any questions on Zoom? No, any final question? Yes, Jenna.
We were a bit sideshow in all of the other stuff that was going on. And so the votes, these 4 votes that were taken were in the last 20 minutes of an 11-day meeting. And they were shoved that late because of this ensnarlment of the text that would be voted on by the membership, because of this very, very deliberate and very effective sabotaging by folks who didn't want it to be passed. So the IUCN as an organization doesn't have an immune system. And so as a virus, you can really screw with them. And these guys were really smart and succeeded at that.